Welcome to our program. I'm John Ankerberg. Thanks for joining me. Our topic is, why are many scientists today rejecting the standard textbook theory of evolution, known as neodarwinism, that we all learn in high school and college textbooks? And where do the problems with contemporary evolutionary theory begin? Well, in the next several weeks, we're going to take a fresh look at the theory of evolution and the scientific problems confronting it with scientist and philosopher Dr. Stephen Meyer. Dr. Meyer is a former geophysicist who received his PhD in the philosophy of science from Cambridge University. He's written 2 best-selling books, Signature in the Cell and Darwin's Doubt. In Darwin's Doubt, Dr. Meyer tells how Darwin himself doubted the ability of his own theory to explain a crucial piece of evidence, and how that doubt has grown up to create a major crisis in evolutionary thinking today. Darwin was troubled by a major event in the history of life known as the Cambrian explosion, in which the first major groups of animals appeared abruptly in the fossil record. Dr. Meyer, we're glad that you're here, and today I want to begin with a clip from Illustromedia's beautiful documentary movie, Darwin's Dilemma, which asks, how could the complex animals in the Cambrian explosion suddenly come into existence? I want you to watch.
An ancient mystery is etched upon these mountains. A story of primordial oceans and prehistoric life, of creatures stranger than fiction, and the controversy that has surrounded them for more than a century. Buried among these majestic peaks are glimpses of an event that transformed the planet in a moment of geological time. Compelling evidence, etched in stone, that challenges long-held assumptions about the origin of animal life on Earth. Today, most paleontologists think that complex animals first appeared on Earth about 530 million years ago, during a geological period known as the Cambrian. But early in the 19th century, little was understood about this seminal event in the history of life. In 1831, the renowned geologist Adam Sedgwick began to excavate the Cambrian rock strata in northern Wales. He was assisted by Charles Darwin, a recent graduate of Cambridge University. For the young Darwin, the fossils embedded in the Cambrian shale were an intriguing curiosity. But at 22, he lacked the perspective to appreciate their full significance. Natural selection, the theory of evolution, and the origin of species all lay years ahead. So he couldn't imagine that the stones beneath him held a mystery he would never resolve. It was a mystery Darwin would ponder into old age and then pass on to future generations. The mystery of the Cambrian explosion.
Now, Dr. Meyer, you've written a best-selling book called Darwin's Doubt. What was the doubt that Charles Darwin had, and what is the Cambrian explosion? Why is it so important?
Well, Darwin had a doubt about an event in the history of life known as the Cambrian explosion. And the Cambrian explosion is the geologically sudden or abrupt appearance of most of the major groups of animals that have ever existed on Earth. And this is a dramatic event in the history of life because it documents the appearance of the first major complex animal forms and the fact that these animal forms appear so suddenly in the fossil record, which was something that was really contrary to Darwin's thinking about the history of life, which was something that he was acutely aware of and which bothered him, as the film said, right into the end of his life.
Yeah. Well, we're going to introduce this topic as we go along, and I want to go to this next clip, and we're going to show how the fossils in the Cambrian explosion came to be found. I mean, this is fascinating for folks. Watch.
More than a century ago, a stunning window to the Cambrian explosion was opened by a series of discoveries made in Western Canada. In 1886, the Canadian Pacific Railroad reached British Columbia and the Kicking Horse Valley. For the first time, eastern and western Canada were linked by a 2,500-mile steel artery that opened the Rocky Mountains to tourists, adventurers, and men of science. Among them was the geologist R.G. McConnell. Earlier in the year, McConnell had heard reports of a shale bed on the flank of Mount Stephen, just outside the town of Field. Railroad carpenters who had explored the area said it was filled with stone bugs. In September, McConnell climbed the mountain. To his amazement, he found unmistakable imprints of prehistoric life on most of the shales in the bed. McConnell was standing in an ocean of fossilized trilobites.
Trilobites are icons for the Cambrian, and there are billions of trilobites high up on the shoulder of Mount Stephen. And one reason for that is that as they grew, they periodically threw off their old skeleton and made a new skeleton. So basically they made many fossils through their individual lives.
McConnell collected hundreds of these fossils and sent many of them to other scientists for examination. News of his work soon reached the offices of the United States Geological Survey and Charles Doolittle Walcott, a leading expert on Cambrian paleontology. On August 30th, 1909, Walcott led his team below this ridge, 15 miles north of Mount Stephen. There, legend holds, he stopped to examine a pile of shale that blocked the narrow horse trail. As he picked up a slab, the geologist noticed a faint but well-defined fossil he had never seen before, a delicate lace crab he later named Morella.
He knew plenty and plenty about the Cambrian. He was an expert on the Cambrian. He published many papers. And when you see this little Morella, it's only about 1 centimetre in length. You get out your hand lens, and you suddenly see that this is, shouldn't be there. This is soft-bodied, effectively. And I'm sure he realised in seconds what it meant. He must have.
All right, Dr. Meyer, why were these Burgess fossils so important?
Well, the fossils of the Burgess Shale documented that the Cambrian explosion was even more explosive than Darwin had realized. Now, Darwin was really troubled by the Cambrian explosion, and just from the fossils that he knew, trilobites and a range of other forms. But he anticipated that future fossil finds would fill in the missing ancestors in the lower Precambrian rocks so that the history of life would look like a great branching tree, as I have on the slide here. But unfortunately, what was discovered from the standpoint of a Darwinian theory was that you had this sudden appearance of all the first complex forms of animal life, but the ancestral forms, the slightly simpler forms in the lower Precambrian strata, were not present. And the Burgess Shale, rather than documenting those ancestral forms, actually documented a range of new complex animals that even Darwin didn't know about. And so, in effect, the discoveries of the Burgess Shale showed that the Cambrian explosion had been even more explosive than Darwin anticipated. And so the mystery of those missing fossils was even more acute.
And the discoveries in the fossil record were just beginning. And as the paleontologists continue to dig, as we're going to see in this next clip, what they found next was astonishing. I want you to watch.
In the summer of 1910, Walcott found a fossiliferous band in the ridge. After blasting a quarry, the geologist and his family unearthed thousands of exquisitely preserved specimens from soft-bodied animals previously unknown to science. He called the site the Burgess Shale.
There in Burgess Shale, especially the lower level which Walcott first exploited, the preservation is miraculous, it's astonishing. We find trilobites, of course, but we find many, many other sorts of arthropods, almost none of which are ever found in a typical Cambrian assemblage. So we can treat them effectively as being soft-bodied. They have almost no chance of being fossilized in normal circumstances.
Geologists believe that the animals of the Burgess Shale were buried quickly and alive by an avalanche of sediment that created an airtight tomb and prevented the decay of soft body parts like eyes, legs, and internal organs.
Now, in the animal morella, very often there's a sort of what we call a dark stain. And I find this very intriguing because that dark stain evidently is the body contents are oozing out. So in other words, the animal is beginning to decay and then something stops. On many of the arthropods we have the most delicate branches, and you can see every single fine hair along them. Quite astonishing. Similarly, the antennae going out like that. In particular instances we have some worms, so we see the outside of the body. We can see various things at the front which enable the worm to burrow through the sediment. But then you look at the animal itself and you can see this sinuous reflective line, and of course you say, oh, that's the gut, that's the alimentary canal. And then in certain cases you actually look at one part of the alimentary canal and you can actually see food inside it. Shellfish which is swallowed. It is a remarkable insight into a fossil you'd never expect to be fossilized.
The Burgess Shale was once part of a massive reef in the Pacific Ocean, a haven for a menagerie of life that thrived at the edge of what is now the North American continent. Throughout long periods of geologic upheaval, tectonic forces elevated these rocks and the fossils they bear more than 7,000 feet above sea level. Here the basic plans of major animal groups that still exist today, and many others now extinct, made their first appearance in the fossil record so suddenly that biologist Richard Dawkins noted, "It is as though they were just planted there, without any evolutionary history."
All right, Stephen, let's tell the folks why we were showing them all this information. Why were the Burgess Shale fossils such a surprise?
Well, they were a major challenge to Darwin's theory, and they challenged Darwin's theory in two different ways. First, they challenged his picture of the history of life. As I mentioned before, Darwin had depicted the history of life as a great branching tree where the animal forms at the top of the tree represented all the forms of life that exist today. In the very base of the tree, there was represented by a single one-celled organism, and then all the branches, connecting branches, represented how that single first organism gradually morphed and changed and became many different things over long periods of geological time. And so the first animal forms, according to Darwin, should have arisen very gradually through a series of small incremental variations and steps. But instead they appear very abruptly. So rather than a tree-like picture of the history of life, the Cambrian fossil record documents something much more like a lawn or an orchard of separate trees that never quite connect at the base. And so you have this sudden appearance and challenges the picture of the history of life. It also challenges Darwin's idea of the mechanism by which living forms would have arisen. His idea was that natural selection, the mechanism of natural selection, would have acted on small incremental variations that would have accumulated over many, many, many generations. And therefore that life would have arisen very, very gradually over time. The first complex animals would have arisen very gradually. But instead what we see in the fossil record is this abrupt appearance of complexity, of complex animal forms, and this is also very contrary to Darwin's thinking about how, the mechanism of how these organisms would have arisen.
Yeah, and part of the evidence, what we're going to see next, is how complex the animals that were found in the Burgess Shale, how actually complex they were. I want you folks to watch.
These fossils gave science its first detailed look at the biology of the Cambrian seas. With computer animation, we can now bring that world to life. Like something out of science fiction, Opabenia was a creature so bizarre it still eludes classification. While its 5 eyes watched for predators, the animal captured its prey with a grasping claw. First described in 1899 from a fossil found on Mount Stephen, Wiwaxia has also puzzled scientists. This mysterious Cambrian animal was covered with overlapping scales and may have fed by scraping microscopic particles off the seafloor. The animal most frequently discovered at Walcott's quarry was Morella. More than 15,000 fossil specimens of this prehistoric crab have been excavated, most revealing multiple pairs of jointed legs and feather-like gill branches used for swimming. The anatomy of hallucigenia has baffled paleontologists since Walcott first discovered its fossilized remains. 2 rows of sharp spines on its back and more than a dozen needle-thin legs gave the animal the appearance of being upside down, even when it was right side up. At the top of the food chain was Anomalocaris, the undisputed terror of the Cambrian seas. Measuring up to 3 feet long, this super predator used barbed feeding arms to capture both hard and soft-bodied animals. It then devoured its prey with layers of razor-sharp teeth. Between 1910 and 1924, Charles Walcott collected more than 60,000 Cambrian fossils, many of which are still studied in museums and research centers around the world. But the treasures of the Burgess Shale represent more than a wealth of information about ancient life. They are also flashpoints in a controversy that began long before the great geologist ever set foot in the Canadian Rockies.
All right, folks, this stuff is astonishing. And before I ask Dr. Meyer to explain why nothing distressed Darwin more than the Cambrian explosion, I first want you to hear what Charles Darwin himself said. Watch.
In 1859, this country estate, 30 miles south of London, was ground zero for a scientific revolution. Here, in the solitude of his study, Charles Darwin completed his landmark book on the origin of species. In it, Darwin attempted to explain how every organism that had ever lived evolved from a single common ancestor as a result of natural selection acting on random variations. Common descent and natural selection became the twin pillars of modern biology, and Darwin's branching tree of life its foremost icon. Yet despite the clarity and detail of his argument, Darwin acknowledged a problem that defied explanation: the Cambrian fossil record.
The distinctness of specific forms and their not being blended together by innumerable transitional links is a very obvious difficulty. I allude to the manner in which species belonging to several of the main divisions of the animal kingdom suddenly appear in the lowest known fossiliferous rocks.
When Darwin was writing The Origin of Species, it was well known at the time that the first fossils of animals appeared suddenly without precursors in the geological record. So there was a deep conflict between what his theory told him to expect to find, namely an abundance of transitional forms going back to that common ancestor for the animals, versus what was there in the fossil record.
Darwin knew that if his theory was true, the older rock strata directly beneath the Cambrian layer should reveal a progression of fossils connecting simple earlier forms to complex animals like trilobites through a trail of incremental steps and failed biological experiments. Such evidence would document the trial and error process of natural selection.
But Darwin says in The Origin, where are these transitional forms? They're not there in the fossil record. What we see instead are fully formed, discrete groups. Now that's a world-class puzzle for someone like Darwin.
If my theory be true, it is indisputable that before the lowest Cambrian stratum was deposited, long periods elapsed, and during these periods of time, the world swarmed with living creatures. To the question of why we do not find rich fossiliferous deposits belonging to these assumed earlier periods prior to the Cambrian, I can give no satisfactory answer.
Dr. Meyer, why were these fossils in the Burgess Shale so challenging to Darwin's theory of evolution?
Well, the fossils of the Burgess Shale, as well as all the other Cambrian fossil beds that have been discovered, raise two mysteries, create two mysteries for Darwin's theory, and they were the source of his doubt about the ability of his theory to explain the whole of the evidence. And the first mystery I call the mystery of the missing fossils, and the film clip there documents it very well, that you have the first major complex animal forms arising in the Cambrian, and as you look in the lower Precambrian strata, you don't find the ancestral precursors to those fossils that you would expect to find if Darwin's picture of the history of life is true. And the second mystery is closely related to it, and I call this the mystery of how you build an animal. How would the evolutionary process build an animal? According to Darwin, the process by which complex animal life arose was the process of natural selection acting on small incremental variations. And Darwin understood that the variations would need to be small and incremental, because if there was a large change in the form of an animal from one generation to the next, that would be something like a birth defect or a deformity, and he understood that those kind of gross changes would always be deleterious or detrimental. So instead, the changes that would occur from generation to generation that would be the source of the evolutionary change over time had to be very small and incremental, which meant that the process needed to take a great deal of time. And instead, what we see in the fossil record is this abrupt appearance without the ancestors and without any evidence of that slow, gradual trial and error process having occurred over time. So the Cambrian explosion raises two mysteries: the mystery of the missing fossils and the mystery of what is the process by which all this
Dr. Meyer,
complexity
Charles
came
Darwin
about.
was a brilliant man. When he was faced with this evidence, how did he try and solve this problem?
Well, he had a charming analogy that he developed that in which he likened the history of life, in particular the fossil record, to a book with many chapters in it. And he said, if we can think of the fossil record as a series of layers where the layers represent just different chapters in the history of life, we may have a situation where we have some chapters that have been lost, and where we maybe only have a page in one chapter, or where there's a complete chapter missing. And so he essentially tried to explain the absence of the ancestral fossils of these Cambrian animals, which he expected on his theory to be there, he explained away the absence of these fossils as a result of the imperfection in the fossil record, that either we hadn't found these fossils yet because we hadn't looked hard enough, or perhaps some layers, for whatever reason, were not preserved. They were eroded away. And so he tried to explain it away that way and analogized it to a book where some of the chapters of the book had been torn out.
Yeah. Well, we've got to acknowledge that in spite of this evidence, the fossil evidence, Darwin did not give up on his theory of evolution. And what he proposed, we want to look at next. So here's our next clip I want you folks to watch.
Darwin was deeply troubled by the Cambrian explosion. He called it an inexplicable mystery. But he wasn't about to abandon his theory, and instead proposed that the animals just looked like they appeared suddenly because he thought that the fossil record was incomplete.
I look at the natural geological record as a history of the world imperfectly kept and written in a changing dialect. Of this history, we possess the last volume alone, relating only to 2 or 3 countries. Of this volume, only here and there a short chapter has been preserved, and of each page, only here and there a few lines.
So Darwin argued, well, perhaps paleontological discovery, digging through the rocks, needed more time, that the transitions were out there, that not enough collecting had occurred, not enough sampling, if you will, of the fossil record on Earth, and given time, those transitions would turn up.
All right, Stephen, if Darwin was waiting for new fossil discoveries to solve the mystery of the missing ancestor fossils, what have the new discoveries in paleontology shown us?
Well, that's the really remarkable thing, and that's what's so significant about the Burgess Shale, the great discovery that was made in Western Canada in the early part of the 20th century. New fossils have turned up, but rather than ameliorating or eliminating the mystery of the Cambrian explosion, they've actually made the mystery more acute, because what the new fossil finds have shown, both in the Burgess Shale and also other fossil finds, in particular one in China, what these new finds have shown is that there were many more Cambrian animals, complex animals, that arose in the Cambrian period than even Darwin knew. And so the explosion was, if you will, more explosive than he understood. And for each of these new forms of Cambrian animal life that arose, each of these were in turn lacking discernible ancestors in the lower strata. So whereas before you had a pattern with a group of complex animals arising, each of which was lacking ancestors in the lower strata, now you have an even wider array of new complex animal forms arising abruptly, again, with each of those new forms missing ancestors. So you're looking at an even more explosive pattern as a result of these new discoveries.
Yeah, and even as you said in your book, okay, Darwin's Doubt, you said even a child can look and see that these animals that just appeared are different, they're more complex, or they're very complex. They're really something to look at.
Well, it might be helpful for your audience to get a sense of just how dramatic this event is in the history of life, because there are about 36 phyla, and a phylum is the largest division of animal classification. So there are about 36 phyla in the history of life. About 26 or 27 of those phyla are fossilized, and of those that are fossilized, fully 20 of the phyla first appear in the Cambrian. There are representatives of those phyla that first appear in the Cambrian period. So this is a big event in the history of life. A lot of the new form and structure, the new body plans that have existed on planet Earth first arrived during that period of time, and they do so very abruptly without clear connections to ancestral forms in the lower strata.
That's why they call it a Cambrian explosion. It's boom, it's there. You know, watch this next clip, because new fossil discoveries have been uncovered. Let's learn a little bit more about the Burgess Shale site. Watch this.
3 decades after Darwin's death, Charles Walcott's historic work in the Canadian Rockies did nothing to fill the gaps in the tree of life or the fossil record. Walcott uncovered the remains of Cambrian animals unknown to Darwin, and each demanded its own unique progression of evolutionary ancestors, a trail of evidence that did not exist in the Burgess Shale.
Walcott realized that the Cambrian explosion of life was an even bigger problem than Darwin imagined. So, in an attempt to defend evolution, he reached back to Darwin's explanation of an incomplete fossil record.
Like Darwin, Walcott thought that the Cambrian explosion was an illusion. He was convinced that the fossils were there. They were just inaccessible to scientific discovery, and he expected that they would eventually be found someplace buried deep beneath the oceans.
For decades, Walcott's hypothesis was widely accepted, but untestable. However, later in the 20th century, new technologies led to empirical conclusions.
Once the oil companies started to drill offshore, they brought up what are called drill cores, and inside the core were hunks of sedimentary rock, and some of those rocks contained fossils, but none of them were made by animals that lived before the Cambrian explosion.
Since the 1960s, scientists have also used radioactive minerals and evidence of changes in the Earth's magnetic field to analyze and date undersea sediments. From extensive surveys, they have created this digital map that defines the age of the seafloor.
We now know that the oldest rocks on the bottom of the seafloor only date back to the Jurassic period, which means that on the standard geologic time scale, they're hundreds of millions of years younger than the rocks below the Cambrian strata.
If you are looking for the ancestors to the Cambrian groups, the last place you would expect to find them is out somewhere on the seafloor. Those rocks are much too young.
So how did the Cambrian animals emerge, and where are the signs of their evolutionary past? Darwin and Walcott both confronted and failed to solve this mystery.
Now, Stephen, if the Burgess Shale didn't solve the problems that Darwin and Walcott confronted, did any subsequent discoveries do that?
There have been some subsequent and very dramatic new fossil finds dating from the Cambrian period and from pre-Cambrian rocks as well. But unfortunately, from a Darwinian point of view, these discoveries also have made the mystery of the Cambrian explosion and the mystery of the missing fossils even more acute. There has been a major find in southern China. The first discovery started coming out in the mid-'80s, and these were just exquisitely preserved fossils. And oddly, the fossils that were discovered in southern China were even more exquisitely preserved, even more beautifully preserved, with almost a photographic realism in the fossil impressions. The other thing that came to light because of this Chinese find was that, again, there were even more new complex animal forms alive at that time than we knew of from either the Burgess Shale or from the earlier fossils in whales of which Darwin knew. So with each progressive discovery in paleontology, our awareness of the degree to which this explosive event was explosive has just increased.
Why is it we're not hearing this in our schools?
Well, it's really hard to say, but most of the textbooks don't even mention the Cambrian explosion, and those few that do mention the Cambrian explosion don't mention that it is a major problem for Darwinian thought. And one of the things I do in the book Darwin's Doubt is I tell the story of the doubt that Darwin had, and I show how that doubt has grown up to become a major crisis in evolutionary theory, not only because it raises this mystery of the missing ancestral fossils, but also because it raises a deeper mystery about the evolutionary process itself. It's a mystery I call the mystery of how to build an animal. What process is there that could build these complex animals and do so in the very limited amount of geologic time that is available to the evolutionary process? It's actually a very big problem.
Yeah, I mean, folks, we are just getting started.
While our knowledge of the Precambrian is far from comprehensive, most textbook accounts include a similar chronology and chain of events. About 3.5 billion years ago, primitive life first appeared on Earth in the form of single-celled bacteria. Over time, these cells gathered into clusters to form blue-green algae that floated on the surface of the oceans. life changed very little for more than 3 billion years. Then on the threshold of the Cambrian period, evidence of multicelled organisms first appears in the fossil record. There is also evidence that near the end of the Precambrian, the oceans were inhabited by jellyfish, sponges, and the mysterious ediacaran fauna.
If you go to immediately before the Cambrian, then actually you find something extremely puzzling, because you get large organisms, large fossils, and these are called the ediacaran assemblages, and they have been one of the great headaches for paleobiology and also for evolutionary biology. Why? Well, because basically some look like animals, but other ones don't look like animals at all.
Some of them look like air mattresses, quilted air mattresses. Others look like a frond. They're not plants, but they kind of have that appearance.
Whether the ediacarans were actually animals or plants is still uncertain. But late in the Precambrian, they disappeared from the Earth. Then, long after their extinction, everything changed in a geological instant. In a spectacular burst of creativity, the basic blueprints for most of the animal kingdom exploded into being. And for the first time, biologically complex structures like compound eyes, spinal cords, articulated limbs, and skeletons appeared on Earth. To understand the speed of the Cambrian explosion, imagine the history of life compressed into a single day.
If we imagine the whole history of life on Earth taking place in one 24-hour period, the current standard estimates for the origin of life put it at about 3.8 billion years ago, let's say 4 billion. So if we start the clock then, our 24-hour clock, 6 hours, nothing but these simple single-celled organisms appear, the same sort that we saw in the beginning. 12 hours, same thing. 18 hours, same thing. 3/4 of the day has passed, and all we have are these simple single-celled organisms. Then at about the 21st hour, in the space of about 2 minutes, boom, most of the major animal forms appear in the form that they currently have in the present. And many of them persist to the present, and we have them with us today. Less than 2 minutes out of a 24-hour period. That's how sudden the Cambrian explosion was.
Since Darwin, excavations on every continent have revealed the magnitude of the explosion of life, an event that was clearly global in scope.
You know, Stephen, what we just watched is astonishing, all right? Why is the explosion of animal fossils on Earth in the Cambrian time period such a challenge to Darwin's theory of how life originated?
The abrupt appearance of animal life in the Cambrian period is really surprising for a couple of reasons. First, it takes such a small percentage of the total of geological history. There's so much innovation that takes place in such a small fraction of time that that's really unexpected. You'd expect on a Darwinian basis that you'd have the mechanism of natural selection, random mutation, gradually producing little incremental changes, and there'd be a kind of a steady increase in complexity over time. But instead we see a very discontinuous or discrete increase, and that increase is confined to a very small percentage of geologic time. Now we might be tempted to think, well, yeah, but still that's the Cambrian explosion is roughly a 10 million year event. That's a lot of time. It's certainly a lot of time on the scale of human history. But what I think few of us realize or recognize is that that 10 million years is actually a blink of an eye biologically as well. It's a small amount of time in relation to the amount of time that the mutation natural selection mechanism needs to work. There's a branch of evolutionary theory known as population genetics, and it's a very mathematical branch of biology in which biologists are able to calculate what they call waiting times. This is the amount of time that you would expect for a given evolutionary change to take place. And biologists can make these calculations if they know something about the mutation rate, the generation time, the size of the populations of the different organisms that are involved. And recent calculations that have been made in the field of population genetics are showing that if you need more than a few coordinated mutations, the waiting times blow up beyond hundreds of millions of years. And so a 10 million year window is a blink of the eye biologically. It's not enough time even to generate a few coordinated mutations, let alone the origin of these extraordinarily complex animals. Trilobites, for example, had compound eyes that actually can be seen in the fossil remains of these animals. These are exquisitely complex forms of visual apparatus that are preserved in the fossil record, and they arise, again, very abruptly and within this 5 million year window. And this is really just not nearly enough time for natural selection mutation to do its work. So it's very surprising and a big challenge to the creative power of the natural selection mechanism.
Yeah, and the mystery only gets deeper as new discoveries are coming in. And we're going to take them in this next clip to China, where just an incredible discovery was found. And what's incredible about this, folks, is that they found older fossils, and they found that these older fossils were more anatomically complex. I want you to watch this clip.
Most recently, several discoveries in southern China have fascinated science and deepened the Cambrian mystery. In 1984, one of the most important finds in the history of paleontology was made outside a small town in China's Yunnan province. While surveying this mountain near Zhenjiang, Ho Xianguan unearthed Cambrian fossils older, more diverse, and better preserved than any ever discovered. The condition of the Zhenjiang fossils was so remarkable, Ho said, it appeared as if the animals were alive on the wet surface of the mudstone.
The fossils they've collected, I mean, first of all, they're stunning. I mean, they're really beautiful to look at.
They're brightly colored, stained with iron and probably other kinds of minerals, so they're kind of golden looking or kind of reddish, and they really stand out from the rather tan-colored background of the rocks, and they're just beautiful. So aesthetically, they're wonderful.
Many of them are soft-bodied. No hard parts, no skeletons, no shells, just soft-bodied, and yet they're exquisitely preserved. So you can see the Cambrian explosion in greater detail than you can anywhere else in the world.
In the early 1990s, reports of the Chinese fossils were released to the rest of the world. At the University of San Francisco, marine biologist Paul Chien followed the news.
What drew my attention was, in fact, a couple articles published in People's Daily, the official paper from the Communist Party in China, announced that the Zhenjiang fossils drew the attention of scientists worldwide. People's Daily reported that this find actually challenges the theory of Darwin's evolution.
Since 1996, Paul Chien has made several trips to southern China to conduct his own investigations.
The Cambrian explosion does challenge the traditional idea of gradual evolution of animals because they all seem to appear all of a sudden, and the problem is, how do we explain it?
Paleontologists have determined that the Chinese fossils were older than those excavated at the Burgess Shale. Yet anatomically, they were often even more complex. This discovery also confirmed that previous estimates of an explosion lasting 20 to 40 million years were much too long.
The time period that we figured it took the animals to be established in the ocean in those days took probably 10 million, 5 million years. So this is truly an explosive event in the scientific terms. What we are seeing is a quantum jump, and this quantum jump has no explanation.
As the interval of the Cambrian explosion is compressed, in other words, as the time available shrinks, the challenge to evolutionary theory grows, because the differences in form that have to be constructed very rapidly are much more dramatic. It's going to pose a real, and I think fundamental challenge to evolutionary mechanisms.
From what I saw, the Chinese scientific community as a whole seemed to be rather progressive around this. They are convinced by the evidence that the Cambrian explosion is real. And they see it's a challenge to the Darwinian theory. They are honest about it. Therefore, they are thinking about how to explain this outside of the Darwinian thoughts.
Now, Dr. Meyer, this stuff is amazing, okay? And given how quickly these complex animals arose and the absence of prior ancestors, how have the neo-Darwinian paleontologists attempted to explain the missing Precambrian ancestral fossils?
Well, as Dr. Chen alluded to, there are many paleontologists now who are acknowledging that the Cambrian explosion is a real event and that it does pose a challenge to the textbook theory of evolution that we all learn called neo-Darwinism. But there are still defenders of neo-Darwinism out there, and what they have typically proposed to explain away the absence of these ancestral forms is something called the artifact hypothesis. And what the artifact hypothesis is a kind of modified version of the idea that Darwin put forward in the 19th century, that we're not seeing the ancestral forms either because we haven't looked hard enough or because for some reason these ancestral forms were not preserved. So the technical way of referring to that is the idea that the missing ancestral animals to the Cambrian animals that arise later, those ancestors are missing, and they're missing as an artifact of either incomplete sampling of the fossil record or as a result of incomplete preservation. Now, 150 years since the publication of The Origin of Species, and given all these different fossil finds, no one really can say we haven't looked hard enough anymore. So what people are now saying is that the fossils are missing because of incomplete preservation. And in particular, the idea that's put forward is that the missing ancestors weren't preserved because they were either too small or too soft to have been preserved. And that's the version of the artifact hypothesis that's still in currency. And yet there's been another discovery in China that has really challenged that idea. In fact, I think challenged it in a very fundamental way.
Yeah, and that's what we're going to go to next in this next clip, folks. What fossil evidence refutes the artifact hypothesis that the reason the missing links, the prior ancestors of the Cambrian animals, have not been found is because they were too soft and too small to have been preserved? I want you to watch this next clip.
The Xinjiang fossils provide the most inclusive picture of the Cambrian explosion yet documented. And directly beneath them, in Precambrian shales, another chapter in the history of life is written in the rocks.
There's another amazing find that's been made in China. Paleobiologists have discovered little tiny microscopic sponge embryos in the layers of rock just beneath the layer that documents the Cambrian explosion.
These embryos were soft-bodied animals. Some fossilized 60 million years before the Cambrian explosion.
They're eggs and embryos, which are preserved in thin crusts of mineralized material, a phosphatic material, on ancient seafloors, which suggests that the chemistry of the seawater in those days was somewhat different than it is today, because this method of preserving fossils disappears during the Cambrian, and it's not around today. So we're lucky that we have these thin crusts with little tiny fossils in them.
This is highly significant because one of the most popular explanations for the missing Precambrian fossils is that the Precambrian animals were too soft and too small to have been preserved.
Since 1999, Paul Chien has studied fossil embryos and helped develop techniques to analyze their structure.
By treating with acid, you can actually remove the rock and isolate the embryos, and then you get round pebble-like or sand grain-like samples. And then we look through some tiny little ones, larger ones up to 1 millimeter in size. And we found about the range between 500 and 800 micrometers. We have mostly sponge. And then I start breaking up these balls and try to start looking inside. And with the help of electron microscope, I was able to see the detailed subcell structure within these embryos.
Jian's work on these fragile remnants of Precambrian life raises an important question.
If these lower strata can preserve an embryo, if they can preserve a soft microscopic embryo, then why couldn't they have preserved the larger ancestral forms that supposedly evolved into the Cambrian animals? In other words, if you can preserve something as fragile as an embryo, why couldn't you, in the same strata of rock, preserve the immediate ancestor of a hard-shelled trilobite?
So the idea that the fossil record is too damaged to provide us with at least a general picture, that idea just doesn't wash.
Stephen, what we just saw is incredible, and I can remember when I was in school having professors tell me, yeah, the missing links are missing because they're soft-bodied, and the fact is there's no way to keep them in the fossil record. That just went down the tubes with that last clip. What are neo-Darwinian paleontologists now saying about what they're going to do?
Well, I think we should just talk about paleontologists and Cambrian paleontologists, because I think increasingly many paleontologists are not necessarily committed to neo-Darwinism because it has so many problems. They're committed to some form of evolutionary theory typically, but now many leading paleontologists, certainly in China, but also leading American paleontologists, are saying that whatever we make of the Cambrian explosion, however we attempt to explain it, we have to start from the premise that this is a real event. It's not an artifact of incomplete sampling. It's not an artifact of incomplete preservation. It's a real event, and it has to be reckoned with. And some of the leading Chinese paleontologists, J.Y. Chen has been very outspoken in making this point. There's a major book on the Cambrian explosion that came out in 2013 by Doug Irwin of the Smithsonian Institution, and James Valentine, who was shown in that previous clip from the University of California, Berkeley, in which they make this same point. The Cambrian is a real event. We may not have a good explanation for it, but we've got to reckon on the event as something that really happened. It's not just something that is a kind of illusion from our not being able to see what happened in the lower strata. We now have good reason to think that the lower strata, or the depositional environment in the Precambrian, was capable of preserving even small and soft organisms, and therefore the absence of the ancestral forms of the larger organisms with hard parts should certainly have been there if those animals existed, if those precursors existed. That they weren't preserved is best explained by the supposition that they weren't there.
Yeah. What does this do to the neo-Darwinian theory?
Well, it certainly, again, challenges the neo-Darwinian picture of the history of life. The first complex forms of animal life seem to arise very abruptly, and it also challenges the idea that the neo-Darwinian mechanism was the mechanism by which these complex forms of life arose, because that mechanism requires a vast amount of time, and it should have left behind a vast number of failed experiments, if you will. It's a trial and error mechanism where you get lots of different variations arising, different mutational changes arising. So we ought to see the evidence of all those failed trials in the lower strata, and they're not there. So it raises questions both about the pattern, the picture of the history of life that Darwin proposed, but also it raises questions about whether the mechanism of natural selection and random mutation is really what was at work in producing these first complex forms of animal life.
Yeah, set us up for next week, because we're going into what you really want to talk about, and that is the fact is we're getting to, in the fossil record, biologists are saying we now know a little bit about how animals are made, and that's a problem.
Well, in my book, Darwin's Doubt, I talk about two great mysteries. And the first mystery is pretty easy to comprehend. It's the mystery of the missing fossils. It's they're simply not there. What we'd expect to see in the in the lower Precambrian layers are not there. But I think there's an even deeper mystery. And that is the mystery that I call the engineering mystery. How do you build an animal? How would the evolutionary process build an animal, especially when you have such a compressed time scale? And that mystery has been made very much more acute by discoveries that have been made since Darwin's time, in particular discoveries in the field of molecular biology about the importance of information digital codes stored in the DNA molecule, and the importance of that information to building animals. And as we've come to appreciate that building animals requires vast sections of digital information, essentially assembly instructions, the Cambrian explosion becomes an engineering problem. Where would all that information come from? How would it get built? Because we now know that you need that kind of information to build an animal.
Yeah. Now, folks, we're just starting to unscramble that for you, and it's a deep mystery, and Stephen is one of the world's best in explaining that, and we're going to attempt to show you what is needed in terms of information, genetic information of how you build an animal, and then we're going to ask the question, where did all of this information come from?
Well, Watson and Crick made the extraordinary discovery in 1953 that is, world-renowned. It was this, they were able to elucidate the structure of the DNA molecule, and they showed that the DNA molecule had this famed double helical structure with 4 chemicals running along the inside of that helix. And as most of us learn in biology class at some point, the DNA is the molecule that contains hereditary information, and it has a helical structure. Along the outside there is this winding ladder, double helix ladder made of sugar and phosphate. On the inside of the molecule there are these 4 chemicals called bases or nucleotide bases. And in 1957, 4 years after the structure was discerned, Francis Crick put forward I think what was one of the most important hypotheses in the history of science. It's called the sequence hypothesis. And what Crick proposed was that these 4 chemicals that chemists represent with the letters A, T, G, and C are actually functioning like alphabetic letters in a written text or like zeros and ones in a section of computer code. That is to say, it's not their chemical shape or structure that matters. What matters is the arrangement of those chemicals such that they are able to convey instructions for building all the important proteins and protein machines that keep cells alive. So what you have on the DNA molecule is literally digital code that provides instructions for building the crucial parts of cells that allow all life to exist.
When you go down that spine, how much information is there?
Well, in the human genome, there's about 3 billion nucleotides. And even in a single one-celled organism, there is enough information to build a minimally complex one-celled organism requires about 400 or 500 proteins. And that's going to compute to several hundred thousand what are called base pairs or individual nucleotide letters in that genetic message.
That all have to be arranged precisely.
They have to be arranged precisely so that the instruction set will direct the construction of these proteins, these various kinds of proteins that are needed to keep cells alive. And proteins are essentially the toolbox of the cell that some of them process information, some of them build structural parts, little miniature machines. We're discovering inside cells there are literally little tiny machines. And there's a form of nanotechnology, sliding clamps and rotary engines and robotic walking proteins. And then some proteins catalyze reactions. These are the enzymes that we hear about. So proteins do all the jobs that keep cells alive and animals alive, but they can only be built if the instruction set is right, and that instruction set is stored on DNA.
Even Bill Gates, when he looked at this, what did he say?
Well, Bill Gates has said that DNA is like a software program, only much more complex than any we've ever been able to create. And many biologists have made similar observations. Famed biotech pioneer Leroy Hood has said very, very directly that DNA contains digital code.
All right, explain how DNA relates to the Cambrian explosion and what we're talking about.
Well, right. I used to ask my students a question, and if you want to give your computer a new function, what do you have to give it? And they'd say, well, code or instructions or software or information, and all of those are correct answers. And it turns out that the same thing is true in life. If you want to build a new form of life from a simpler pre-existing form of life, you need new information, or rather, to put it more precisely, the evolutionary process would need to generate new information, new code. And that raises a big question. How does it do that? How would it do that? How could it do that? And one of the reasons that's such a big problem, a big question, is that the mechanism, the evolutionary mechanism, the driving force of the evolutionary process is thought to be natural selection acting on random genetic mutation, random changes in the arrangements of A's, C's, G's, and T's, the digital characters in the DNA molecule. Mm-hmm. But what we know from experience of computer code, for example, is that if you start making random changes to digital characters in a message-bearing sequence, you're going to degrade the information that's present in that sequence long before you're ever going to generate something fundamentally new and useful. I mean, just ask yourself a question if you're a computer programmer. If you've got a functioning computer program and you start randomly changing zeros and ones, are you going to generate a new program or operating system? Or are you're going to introduce glitches and bugs into the program you already have?
Yeah, all our techies in the room are saying, keep the bugs away.
Yeah, keep the bugs away. And so there's something fundamentally disquieting from the standpoint of information science about the idea that random changes in a functional section of code or text could generate something fundamentally new. And so that's been a kind of general concern that scientists have had about the creative power of the Darwinian mechanism. Can the Darwinian mechanism of natural selection and random mutation really generate new functional code? Because to build a new animal, you need new code. You need new DNA with digital information stored in those molecules.
All right, let's go to the clip where you show what is happening in the cell and how it's programmed with this code.
How do you explain the origin of the Cambrian animals seemingly out of nowhere? This isn't just a problem of explaining the absence of evidence in the fossil record. It's also a problem of explaining everything we know about life, right down to the level of molecules and cells.
The biological structure of a Cambrian trilobite was as complex and sophisticated as a modern crab. Its organs included a brain, gut, heart, and compound eyes. Each organ was constructed from specific types of cells. Each cell type was made from dozens of specialized protein molecules. And each protein was assembled from a 4-letter chemical code in a section of DNA called a gene.
Now, for the evolutionary process to transform a simple Precambrian organism like a sponge with 4 or 5 cell types into a Cambrian trilobite with at least 10 times that many different types of cells, that's a huge leap in complexity. And to make that leap, you need a vast amount of new genetic information. Where does that information come from? That's the central mystery of the Cambrian explosion.
So, Dr. Meyer, help us understand this. Go back to this thing now of chance. I think Murray Eden at Worcester Institute called a conference in the 1960s, and he was a mathematician, and he called evolutionary biologists, and he had people that were working on the atomic bomb. He had all kinds of scientists there, and he had a question that was bugging him. What was it?
Well, Murray Eden was a computer scientist at MIT, and there was a picnic in 1965 where the computer scientists and the physicists and the mathematicians and the engineers were talking with their biology colleagues over lunch. And all these scientists with mathematical training were expressing skepticism about the creative power of the mutation selection mechanism. They didn't believe that it could generate new genetic information on the scale that was required to explain things like the Cambrian explosion. And their main reason for thinking that was they realized that random changes in any formal language system will inevitably degrade meaning. And that is because there's a lot more ways to go wrong than there are to go right. If anyone's played Scrabble and you have a pile of letters, you know that if you just reach in and start picking the letters out at random, you're overwhelmingly more likely to get gibberish than you are to get a word. And that's actually because people have worked out the math on this. The ratio of functional words for any given length to the ratio of sequences of characters that give you gibberish is really minuscule. It's in the case of a 12-letter word, I think the ratio is 1 over 100 trillion. One word for every 100 trillion arrangements of characters that give you nothing but gibberish. So if you start with a word or start with a section of functional genetic text or a section of computer code and you start randomly changing it, you're overwhelmingly more likely to end up with gibberish because most of the sequences are gibberish. And that's the problem that the engineers were concerned about at Wistar. And so this is just not a good way to think about how genetic information originated. This mechanism simply is unlikely to produce anything good in the time we have available.
Or as Murray Eden says, it's improbable. It probably couldn't happen.
It's improbable on a cosmic scale. Yeah.
All right, let's go to the clip where you show what is happening in the cell and how it's programmed with this code.
In 1957, Francis Crick first proposed that chemicals called bases along the spine of the DNA molecule function as alphabetic characters in a written language or digital characters in a machine code. This animation shows how this digital information directs protein synthesis. First, a large protein complex separates the tightly wound strands of the DNA to prepare it to be copied. During this process of transcription, a protein complex called a polymerase produces a single-stranded copy of the original instructions. Here we see this copy, a messenger RNA molecule, being constructed inside the polymerase as individual bases are positioned and added to the growing strand. Now we see the polymerase in action from the outside as it spits out the messenger RNA transcript. Next, this RNA transcript approaches and passes through a molecular machine called the nuclear pore complex, an information recognition device that controls the flow of information in and out of the cell's nucleus. Now we see the genetic assembly instructions on the messenger RNA approaching and arriving at a two-part chemical factory called a ribosome, the site of protein synthesis. As the messenger RNA transcript passes through the ribosome, the process of translation begins. During translation, a mechanical assembly line builds a specifically sequenced chain of amino acids in accord with the instructions on the transcript. These amino acids are transported from other parts of the cell by molecules called transfer RNAs, which link specific sequences of bases to corresponding amino acids. The sequential arrangement of the amino acids determines the type of protein constructed. When the construction of the chain is complete, it is transported to a barrel-shaped machine that helps fold it into the precise shape required to perform its function. After the chain is folded into a protein, it is released into the outer cytoplasm to do its job in the cell.
Now, Stephen, looking at that wonderful clip, I bet you that 85% of our audience didn't understand what they were looking at. But they did pick up a couple things. First of all of that was happening in one single cell. All right? All of those little machines that you were looking at are in one little cell. They're interconnected. Then they saw code. Your point is, if that code's not right, those machines aren't going to do their jobs, because that code, that information, makes them do what they do. And the question you're raising is, where did this complex code come from?
Well, right, and what we've understood since the time of Watson and Crick, and from what's called the period of the 1950s and 60s, which we now refer to as the molecular biological revolution, is that information is running the show inside living systems. It's a little bit like what goes on at the Boeing plant up in Seattle where I live, where the engineers use a technology called CAD/CAM, computer-assisted design and manufacturing, where an engineer will sit at his or her console, write some code for building a particular part for the airplane at an assembly center, and the code will go down a wire. It will be translated into another machine code that can be read by the assembly center, and then, for example, if you're building an airplane wing, the assembly arm may take the instructions and put the rivets at exactly the right place in accord with the engineer's instructions. So you have digital code being written by an engineer directing the construction of a mechanical part. And inside cells, what we have is digital information directing the construction not of airplane wings, but rather of proteins. And proteins, again, are the toolbox. They're the molecules that do all the important jobs inside cells that keep us alive. So the recognition that information is running the show inside living systems has also made us aware that if you want to build a new form of life, like a Cambrian animal, you've got to have a lot of new information. Every new Cambrian animal required a bunch of new dedicated cell types, Each type of cell required dedicated proteins. Each new type of protein requires, in turn, new code. So the Cambrian explosion isn't just an explosion of new forms of animal life, it's an explosion of information. And yet there are really big reasons to doubt that the neo-Darwinian mechanism can generate that new information. And in my book, Darwin's Doubt, I go into, I explain the math as to why it's so incredibly improbable to think that a random search for new sections of functional genetic text could produce, even on the scale of the entire history of life, even one new gene or protein.
Stephen, illustrate that with your story about the thief trying to break into a lock.
Let me give you a homespun illustration. Imagine we've got a bike outside the building here, and there is, but it's locked with a nice 4-dial bike lock. Is it more likely that a thief will be able to open the lock as a result of a random search for the combination, or is it more likely that the thief will fail? Now, in a way, it's a trick question, because it depends on how much time the thief has to try to open the combination. We know that there's a lot of different combinations. In fact, with a 4-dial lock, there's 10 times 10 times 10 times 10, or 10,000 possible arrangements of those digits, only one of which will open the lock. If the thief only has a couple minutes, the thief will, by random means, only be able to sample a very small number of those combinations, and so it's overwhelmingly more likely in that case that the thief will fail. But we could imagine a situation where, say, we gave the thief several days to do it, and he was able to sample more than half of the combinations, in which case it would become more likely that he would succeed than he would fail. But what if we change the illustration a little bit? What if we give the thief that same, say, 24-hour period, and now we confront him with this lock, a lock with 10 dials on it? Now, well, with 10 dials, you're not just adding combinations, you're multiplying by 10 with each new dial. So for a 10-dial lock, you actually have 10 to the 10 possible combinations, or 10 billion combinations. So even in a day, a full 24-hour period, the thief is only going to be able to sample a very tiny fraction of that total 10 billion. So in that case, it's overwhelmingly more likely that a random search will fail to find the meaningful combination that will open the lock. Mm-hmm. Now, the question in biology is the situation for finding a new gene or protein more like the first case, where the lock has a reasonably small number of combinations in relation to the time available, or is it more like the second case, where the number of combinations is so vast that even with a great deal of time, there's not going to be enough time to sample more than a small fraction of the combinations?
And what is it?
Well, it turns out that the biological system confronting us is not like the 4-dial lock with the day, it's much more like the 10-dial lock. In fact, there's a scientist named Douglas Axe who's given a very precise estimate of this, and he has shown that that it's much more like a bike lock with 77 dials, which is one for every functional arrangement of those letters in the DNA code. There are 10 to the 77th arrangements that will give you gibberish, that is a non-functional, non-folding amino acid chain rather than a functional protein. And so, with that many combinations to search, even if you have 3.5 billion or 4 billion years of biological history to do the searching, by a random means, you're not going to have enough time to sample but a small little fraction of the different combinations. And so it's going to be overwhelmingly more likely that a random search, one relying on random mutation, will fail to find even one functional gene or protein in the known history of life on Earth. That's the upshot of Axe's calculations. And I go into this and explain this in more detail in the book Darwin's Doubt.
Yeah. We all kind of grew up in public schools and were taught that evolution, life arose by chance. Okay? But I can hear some folks saying, you know, there's a small probability that this could have happened, so why isn't it possible?
Well, people will look at a number like 1 chance in 10 to the 77th power, and they'll say, so there's a finite probability that a new gene or protein could arise in the known history of life on Earth. And there's a flaw in that reasoning. We can never say anything is impossible because there's always a finite probability of any event happening. But the question is whether or not this is a good explanation or a bad explanation. And I have an illustration for why this is a bad explanation. There's a famous comic named Jim Carrey who, in a particular movie, is playing a character who's kind of a goober, and he approaches a young lady that he fancies, and he says to her, what are the odds that a girl like me and a guy like you could get together? And he botches his pickup line, and she says, well, not good. And he says, what do you mean not good? Like 1 in 100? And she says, no, like 1 in a million. And he starts jumping up and down. And he gets all excited. He says, oh, so there's a chance. There's a chance. And I sometimes think this is the way our Darwinist friends reason, that just because there's a finite probability doesn't mean that the event is at all likely to happen. And in the case of the idea that random mutations have generated the new genetic information that we need, it's overwhelmingly more likely that such a mechanism will fail than it will succeed. And so the hypothesis that such a mechanism succeeded is actually overwhelmingly more likely to be false than true. So in science, we don't want to choose a hypothesis that's more likely to be false than true. We want the best and most likely explanation. So clearly, we should be looking someplace else other than the Darwinian mechanism to explain the origin of information. Yes, there's a finite probability, but it's overwhelmingly more likely that that is not how it happened, and therefore we should be looking for a better explanation.
The biological structure of a Cambrian trilobite was as complex and sophisticated as a modern crab. Its organs included a brain, gut, heart, and compound eyes. Each organ was constructed from specific types of cells. Each cell type was made from dozens of specialized protein molecules. And each protein was assembled from a 4-letter chemical code in a section of DNA called a gene.
Now, for the evolutionary process to transform a simple Precambrian organism like a sponge with 4 or 5 cell types into a Cambrian trilobite with at least 10 times that many different types of cells, that's a huge leap in complexity. And to make that leap, you need a vast amount of new genetic information.
Where does that information come from? That's the central mystery of the Cambrian explosion.
According to neodarwinism, new proteins are constructed by the dual mechanisms of genetic mutations and natural selection. As the genetic instructions for building proteins are copied, an occasional error can alter their contents. If these accidental revisions prove beneficial to survival, they are selected or preserved and passed on to future generations. Over eons, these small changes accumulate, and new proteins, cell types, and even Cambrian carnivores gradually evolve into existence.
Richard Dawkins, the famous Oxford evolutionary biologist, has illustrated how the Darwinian mechanism works using a metaphor he calls climbing Mount Improbable.
From the front side, the mountain is a sheer cliff that could never be scaled in one giant leap. For Dawkins, this represents the impossibility of creating a complex animal by chance alone. Yet Dawkins also envisioned an alternative route up the backside of Mount Improbable, a long, gradually sloping trail of small steps leading all the way to the summit.
According to Dawkins, that's how you'd climb the mountain, and that's also how you'd build a Cambrian animal, one small step at a time. What chance alone can't accomplish in one blind leap, natural selection can accomplish through the cumulative effect of many small incremental steps.
In theory, each step corresponds to a small unit of biological change, a new gene and its protein product. But do mutations and natural selection have a reasonable chance of producing even one protein in the time available? Since 1992, molecular biologist Doug X has examined this question.
There's a story that's being told, and there's a appeal in the case of Darwinism to random mutation and natural selection as being, in vague terms, the mechanism. But if you look at the detail, what kind of mutation can accomplish these transitions? And there, it's important to realize that the one area where we can really nail this down is at the single protein level, where you can actually measure it. And if you look at protein structures, to get a substantially new protein fold is prohibitively difficult.
You know, Dr. Mayer, after watching that clip, Richard Dawkins and other evolutionary biologists are still confidently proclaiming that it's possible to build a Cambrian animal by natural selection and random mutation in small incremental steps. But what are some of the problems that you see with this evolutionary view?
Well, actually, many of the leading Cambrian paleontologists are now disputing that view. There's a paleontologist at the Smithsonian named Douglas Irwin, who has written a book with James Valentine from the University of California, Berkeley. And they take what they call a non-uniformitarian evolutionary view, which just in plain English means that whatever caused the Cambrian animals to emerge, it's unlike anything any biological process we see at work today, including the mutation natural selection process. So there's a lot of scientists who are doubting the creative power of the mutation selection mechanisms. One of the reasons for that is that natural selection can only select for a functional advantage, for an advantageous change in a protein or a gene at the smallest level. And yet we know that the odds of generating such a new gene or protein by random mutations alone is extremely small. And it's small even when we take into account the number of opportunities there are for such an event to occur, given the known history of life on Earth. So the idea that mutation and selection have this amazing creative power is really increasingly being questioned. It does a great job of explaining small, minor variations in existing protein folds and structures. But if you need enough changes to build what's called a new protein structure, a new protein fold, too many things have to go right before there's any selectable advantage. There is no gradual series of steps up the backside of the mountain. It's basically a big jump proposition where you need many coordinated changes to occur before there's any selectable advantage. And the number of coordinated changes that are required are so vastly improbable that it's far more likely, as we were saying in the last program, that such a mechanism will fail. Such a random search will fail than it is that such a mechanism will succeed. So there are really important, I would say, mathematical reasons to doubt the creative power of the mutation selection mechanism with respect to the problem of building even one gene or protein, let alone a whole new Cambrian animal, which would require hundreds and thousands of new genes and proteins.
Well, let's show them a clip that illustrates this. This next clip explains why Darwin's theory of natural selection and random mutation cannot account for what is needed to build a Cambrian animal. Folks, I want you to listen.
Each of the thousands of different proteins in nature is actually a chain made from a specific combination of 20 different amino acids. The sequential order of these chemical building blocks is crucial, for if they are arranged correctly, the chain folds into a functioning three-dimensional molecule. But if the amino acids are incorrectly assembled, no protein will form. If proteins are indeed rare among the possible sequences of amino acids, what are the odds that mutations would stumble upon a functional combination of chemicals from the vast number of alternatives? To find out, Axe randomly altered the structure of an enzyme protein comprised of 150 amino acids.
You've got the protein 150 amino acids long, then you've got 20 to the 150th power possible ways of arranging the amino acids. Out of all those possibilities, how many are functional and how many are gibberish?
If you do the experiments and you analyze how much information is required to get, say, a new protein fold, it's just far beyond what you can get by random mutation and natural selection.
How far beyond? Axe published his findings in the Journal of Molecular Biology. He determined that among all the possible amino acid combinations, the probability of generating just one short protein by mutation is roughly 1 in 10 to the 74th power, or 1 chance in 100 trillion, 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 trillion.
To put that in context, there's only 10 to the 65 atoms in the entire galaxy. So to build a new functional protein by selection and mutation within the time allowed for the Cambrian explosion
what you're essentially having to do is equivalent to a blindfolded man looking throughout the entire galaxy for one marked atom. So what we're talking about is searching for a tiny, tiny needle in an enormous haystack and having a very limited time to search.
So on the question of something like the Cambrian explosion, there does not appear to be any way that unguided random mutations can accomplish what needs to be accomplished to explain new functional proteins. And certainly by extension, wherever in the history of life you would need to have multiple new protein folds, the probabilities multiply. So there's no reason to think that this is plausible.
Dr. Meyer, that's really impressive. Do you have another way of illustrating what we just saw?
Well, we talked in the last episode about a bike lock as an illustration. You have a thief who wants to perform a random search to find a combination in order to steal a bike. He comes up against a big hurdle, and that is the number of combinations that have to be searched. In a 4-dial bike lock, you have 10 digits on each dial, so you've got 10 times 10 times 10 times 10, or 10,000 possibilities that have to be searched. And but depending on how much time is available, it could be either be plausible that the search will be successful randomly, or implausible. What Axe has shown is that in the case of even a single gene or protein, Douglas Axe, the scientist that we saw in the last clip, is that in the case of even a single gene or protein, the number of dials that we are effectively looking at is about 74, there's slight differences in his estimates depending on the method he uses, where you've got 10 possibilities at each dial. So that's an enormous number of combinations that correspond to that. That's 1 in 10 to the 74th power combinations. Now, that number is so big, it's many orders of magnitude greater than the number of atoms that exist in the Milky Way galaxy, that you can see that you would have to be looking a long, long, long time by random means to sample more than half of those possibilities. Once you get beyond half, then you could say it's more likely that you'll succeed than you will fail. But because the number is so big, there's so many possible ways of arranging those amino acids or the letters in the DNA code that would correspond to building those proteins, that it's even on the scale of cosmic time, or on the scale of the history of life on Earth, you're going to not have nearly enough time to sample but a tiny fraction of that total number of combinations, which means it's going to be overwhelmingly more likely that a random search will fail to find even a single functional gene or protein in the known history of life on Earth than it is that such a random search will succeed. And if it's overwhelmingly more likely that a random search will fail to find a new gene or protein, fail to generate new genetic information, then the hypothesis that such a search succeeded is actually more likely to be false than true, in which case that's a bad hypothesis and we should reject it.
In this next clip, we're going to show the problem only gets worse, okay, because scientists have discovered something else. And this was amazing when I watched this. They discovered the body plan for an animal does not reside in DNA. So where is the information of each animal body plan stored, and where did that information come from? Folks, I want you to watch this clip.
But the inability of random mutations to generate new genes and proteins is only part of the problem. For the origin of Cambrian body plans demanded more than new genetic information.
A lot of the information for specifying an anomalocaris, a trilobite, what have you, it does not reside at the DNA level.
The body plan, as far as we know, is not in the DNA.
While DNA carries the instructions to manufacture proteins, it cannot alone assemble them into cell types, or arrange cell types into new tissues and organs, or tissues and organs into body plans. Instead, the formation of body plans ultimately requires another level of information stored somewhere in the three-dimensional structure of the egg cell and the embryo. Instructions that direct the development of complex animals from fertilized eggs. With computer animation, we can observe this intricate process. As an egg cell begins to divide and differentiate, a network of biological commands orchestrates the development of an arthropod. After several stages of division, dozens of new cells align against the outer membrane of the egg. And then, cued by a chemical signal, they start their migration toward targeted areas in the embryo, where they will gather and develop into a mature organism. The cells steadily increase their numbers and align, like members of a marching band, into patterns that will form the tissues and organs, head and legs of the growing embryo.
That happens by a process of cell specification and differentiation, where cells are committed irreversibly to performing particular roles. You're going to give them different jobs to do. You're going to be part of the locomotory system of this organism. You'll be an eye, you'll be a gut, and so forth. To me, that's an absolutely astonishing process, but it works. And what it builds you is different kinds of organisms depending on the instruction set that's provided.
So there is an organismal blueprint, an ultimate point that the embryo hones in on and is attracted to and eventually embodies.
That foresight, that preordained outcome is built into the embryo.
When you talk about these early developmental sequences, and anomalocaris and opavinia, what have you, you're talking about information in the broad sense, codes, specifications, entailments, implications that are orders of magnitude beyond anything that we can currently conceive. It's so off scale that you've left that line of impossible by chance a long time ago.
The volume and complexity of information that controls the development of a body plan is staggering, and its location in the cell stands as perhaps the ultimate challenge to the neo-Darwinian scenario of random mutation and natural selection.
We know that much of this higher level information that's required for building new tissues and organs and body plans isn't found in DNA. That means that you can mutate DNA indefinitely without respect to probabilistic limits, without respect to time and number of trials, and you're never going to get the kind of form and structure you need to build a new organism. DNA is simply the wrong tool for the job, and no amount of time is going to overcome that limitation. That has a really devastating implication for the neo-Darwinian mechanism.
If the Darwinian mechanism cannot explain the origin of the information necessary to produce the Cambrian animals, is there any other cause that can?
So, Dr. Meyer, help us understand this a little bit more.
This last problem that we saw explained in the film is really a very profound and fundamental problem in modern biology. According to neo-Darwinism, new form arises as a result of new variations, and the new variations occur as a result of mutations in the DNA. So the ultimate source of new biological form are changes in the arrangement of those A, C's, and G's in the DNA molecule. But what developmental biologists are learning is that DNA is only part of the story. It provides a necessary amount of information for building proteins, but that information is nowhere sufficient to build a whole animal, because the proteins have to be arranged into structures within cells. Different types of cells have to be arranged to form tissues. Different tissues have to be arranged to form organs, and organs and tissues need to be arranged to form whole body plans or architectures, where a body plan is a unique arrangement of body parts and tissues. So DNA is necessary but not sufficient. Now, if we think of that in light of what neo-Darwinism says, it says that the source of new form and structure in the biological system, a new body plan, is going to come from variations in the DNA.
But if the DNA only provides the information for building proteins, and other levels of information are necessary to arrange the proteins, the cell types, the tissues, and the organs into a whole body plan, then you could mutate DNA indefinitely, and you will never build a new organism. The technical way of saying that is if body plan building or body plan morphogenesis depends on information beyond DNA, then mutating DNA is never going to get you a new body plan. It's not the right tool for the job. Now, I first learned about this in a technical book published in 2003 by MIT Press that was co-edited by two leading evolutionary biologists. And in the introductory article to this book, in which they explained this problem of the higher levels or the epigenetic information, they acknowledged that neo-Darwinism was still the standard textbook theory, but they said that neo-Darwinism has no theory of the generative. That is to say, it can't explain the big changes in the history of life. It doesn't explain the origin of body plans. Now, one of the things that struck me about reading that book and the many articles that were elucidating the difficulty of this problem is that there's a huge disparity between the public presentation of the status of evolutionary theory as we find it in our biology textbooks, as we find it presented by people like Bill Nye the Science Guy, or by Richard Dawkins, or by defenders of the Darwin-only approach to science education on the one hand, and the actual status of evolutionary theory as we find it in the peer-reviewed technical literature in evolutionary biology, where many evolutionary biologists are now saying very openly that neo-Darwinism is dead and we need to find a new theory of evolution. And so one of the things I did in my new book, Darwin's Doubt, is first of all, I exposed this disparity and showed that the popularizers are out of date and what they're telling the public is simply false. And I also looked at the many new theories of evolution that are being proposed to replace neo-Darwinism. I examined them and then also critiqued them and showed that they too fail to explain the origin of the genetic information in DNA that you would need to build Cambrian animals, but also this higher level epigenetic information. And so there really is a crisis in evolutionary theory today, one that started way back in 1859 when Darwin honestly acknowledged the problem of the Cambrian explosion. But in the book I show that that problem has become much deeper and more profound as we've learned more about what it takes to build an animal. We need both genetic and epigenetic layers of information, and there is no evolutionary model today that accounts for the origin of that crucial information and therefore the origin of these Cambrian animals.
All right, Stephen, I want to talk about the positive side of this. We've been talking about the negative side. What's the alternative that we can go to?
Well, I'm a proponent of the theory of intelligent design. I think what we're seeing in this digital code in DNA and in this hierarchically organized information processing system, which is what an animal is, I think we're seeing positive evidence of a designing intelligence
fundamental profound problems. In the mid-1980s, there were a couple of scientists, Christian Nusslein-Volhard and Eric Wieschaus, who won the Nobel Prize for some important work that they were doing mapping the genome of the hapless fruit fly.
Yeah, I think it's an interesting story.
And what Nusslein-Volhard and Wieschaus did was they effectively reverse engineered the genome of the fruit fly. They would knock out particular genes and then they would see what would develop or what wouldn't develop. So they'd knock out a particular gene and they'd find that that would affect eye function or cause it to diminish, or they'd knock out another gene and it would find that it would deform the wing or something. So they were able to figure out which parts of the genome affected the different parts of the animal as it developed. And for this work, they got a Nobel Prize. It was a fantastic piece of scientific investigation. In addition to mapping the fruit fly genome, these two scientists made a discovery that has profound implications for evolutionary theory. They found that if the mutations they induced were induced early in the development of the embryo as it moved from embryonic form to adult form, that invariably the development of the animal would shut down. And they called this the problem of embryonic lethals, because in fact the developing fruit fly would just die if there were mutations induced early in the development of the animal. Now your viewers may remember from biology class that as embryos develop, they'll go through a pattern of cell division where there'll be the fertilized egg. It will divide into 2 cells, into 4, into 8, and so on, and eventually thousands of different cells. Now, if you wanted to build a new body plan, you would have to induce mutations early in the process, Because if the mutations occur very late in the development, they're only going to affect a small cluster of cells as the animal is developing through all these various divisions. So it's only the cells at the very beginning which can possibly affect the whole architecture of the animal itself. And yet these two German scientists found that inducing the mutations early always caused a termination of the pattern of development.
Which is kind of serious.
Which is really serious, because the very kind of mutations you need to develop a new body plan in the process of evolution are the very kind that we know always kill the animal. So the kind of mutations you need are early acting, body plan affecting mutations which are also beneficial, and they never occur. We get early acting body plan affecting mutations, but they're never beneficial. They're always lethal. The kind we don't need, mutations that occur much later, that affect only a small cluster of cells, those may be beneficial, but they would never change a body plan. So there's a kind of a catch-22. The kind you need, you don't get. The kind you get, you don't need. And this is a big problem for the Cambrian explosion.
I would say it's a real big problem.
Well, it's a problem for explaining the Cambrian explosion, because the Cambrian explosion is, after all, the event in which we get the origin of new body plans, and yet the Darwinian mechanism doesn't seem to be able to produce a change on that scale of innovation.
But that's not the only problem that you've found. What's another one?
Well, there's a related problem, and it's also emerged from studies in the field of developmental biology. There's a famous developmental biologist at the California Institute of Technology named Eric Davidson. And Davidson has mapped what he calls developmental gene regulatory networks. And I have a picture of one on the slide. And these are essentially integrated circuits or networks of genes and their protein products that are functioning like integrated circuits. And the way to understand what they do is this. You have a gene that will direct the construction of a protein, which is actually a signaling molecule. And that protein will then either turn on or turn off the expression of other genes. And those genes will also produce still other signaling molecules will turn on or turn off still other genes. And there's this whole vast network of genes and their protein products that are linked together that have the effect of controlling how cells develop and differentiate from each other during the pattern of animal development. So to build an animal, you need these developmental gene regulatory networks, or these integrated circuits of genes and their gene products. Now, Davidson has discovered something else that's very important about these developmental gene regulatory networks, and that is you can't change them very much, or else animal development shuts down. He says you can't perturb them, or else you again get the problem of lethals. So that raises a profound question for evolution, or for evolutionary theory. Because to build a new animal body plan, to build a body plan, we know we need these gene regulatory networks, which implies that to build a new animal body plan, you would need one gene regulatory network to evolve into another gene regulatory network. But we know experimentally the one thing that cannot happen is for these gene regulatory networks to change very much. So it really raises the question of how you would ever get a new body plan to emerge from an older body plan at all. How would you make that transformation when the very thing you need to transform cannot be changed, namely these gene regulatory networks?
Yeah, and looking at your picture of what a gene regulatory network looks like, it's awfully complicated. I would think what raises the question, too, where did the whole thing come from in the first place?
Well, it's not just complicated. There's an integrated complexity here that's characteristic of circuitry. This is the kind of thing that engineers design. It looks like a motherboard or an integrated circuit. And so it also, I think, raises the question of design. And it does that for another reason as well, because gene regulatory networks are made of a vast network of genes. And each gene contains digital code. And that gets back to this really fundamental question that we've been talking about in previous episodes, is where does all that information come from? We know that we need additional layers of information beyond what's in the DNA, but these gene regulatory networks contain genetic information in a digital form stored in DNA, and that's a big problem too. So at every level we have difficulties that the modern neo-Darwinian synthesis has just been unable to solve. It does not provide an explanation for the origin of the genetic information. It doesn't provide an explanation for the origin of the higher level epigenetic information, and mutations do not give a good explanation for the origin of body plans. We don't get early acting developmental mutations that are beneficial, and yet we need those kind. And we can't perturb these gene regulatory networks, and yet we know that to build a new animal body plan, you would need to generate new developmental gene regulatory network. So there are multiple problems confronting the neo-Darwinian mechanism, and yet that is still the standard mechanism that is taught and proposed as the explanation for the origin of the kind of large-scale change that we see arising in the fossil record in the history of life in events such as the Cambrian explosion.
Yeah, I mean, it raises the question, if, boom, you have all of these animals in the Cambrian fossil record showing up, okay, and all of this stuff is basically there, where did it come from?
Where's the genetic information come from? Where does the higher level information come from? And how do you build a body plan? None of these problems have been answered by modern Darwinian theory, and so we clearly need a new theory. Well, in the mid-'80s, I was a young scientist, and I was working for an oil company as a geophysicist, and I was working in seismic processing, which was an early form of information technology. And a conference came to town in Dallas, where I was working at the time, that was just absolutely fascinating to me. I'd long been interested in the big issues in science, the intersection of science and philosophy. The conference discussed three big questions: the origin of the universe, the origin of life, and the origin and nature of human consciousness. At the conference, there was a scientist there named Charles Thaxton, who had just written a major book on the question of the origin of the very first life. It was called The Mystery of Life's Origin, and he showed that this was an unsolved problem in modern biology, and that there was no chemical evolutionary theory that explained how we got from the chemicals in the prebiotic soup to the first living cell. And the key problem that he focused on, that many of the other scientists on the panel acknowledged, was the problem of the origin of information, the problem of the origin of the genetic information stored in DNA that you need to build the first cell. Well, I was fascinated with this, and ended up getting to know Thaxton, was introduced at the conference, and started to visit with him after work. He had, in the epilogue to his book, The Mystery of Life's Origin, floated an idea that he called, well, he didn't really have a name for it. He said that there was something about the information-bearing properties of DNA that suggested that perhaps we were looking at evidence of an intelligent cause, is the way he put it. And at the time, he didn't develop this into a full-blown theory, but it was more of an intuitive connection based on our understanding, or his understanding, and that of many people, that information is a kind of mind product. It's the sort of thing that comes from a mind or an intelligence or a conscious agent. It's not something that comes from chemistry. And anyway, I got fascinated with this connection between information and intelligence, and wondered if it was possible to formulate the case for what we later called intelligent design as a rigorous scientific hypothesis. And a year later, I found myself heading off to graduate school in Cambridge. And maybe ironically, but it was the case, I started to study very intently the works of Charles Darwin. Of course, I was interested in Darwin because of his ideas about the origin of species and so forth, but I was also interested in his work because he had pioneered a unique method of scientific reasoning that allowed scientists to investigate events in the remote past. And I realized that however we explain the origin of life, whether we invoke design or just undirected material processes, we would be trying to explain something that happened a long time ago in an event in the history of life. So the method that Darwin developed seemed to be really important to deciding whether or not you could develop a scientific case for intelligent design. And the method he developed has a name. It's called the method of multiple competing hypotheses, or it's sometimes called the method of inference to the best explanation. Mm-hmm. And that raised a key question. What does it mean to be a best explanation? And as I got deeper into the subject, I began to study one of his key mentors, which was the geologist Charles Lyell. And one day a light came on for me as I was reading Lyell's book, Principles of Geology, being an attempt to explain the former changes of the Earth's surface by reference to causes now in operation. And that's where the light went on. Because what Lyell was actually saying, as I got deeper into his work, is that the job of the historical scientist is to explain events in the remote past by reference to causes now in operation, causes that we know from our experience are capable of explaining the event in question. So here's an illustration. If you go to eastern Washington today, there's still places out in the Palouse country where there are thin dustings of white powdery stuff. And if you're a geologist and you don't know what happened on May 18th, 1980 when Mount St. Helens blew up, and you're looking at that white powdery stuff, you're going to use the method of Lyell and Darwin. You're going to generate multiple hypotheses. Maybe it was a flood. Maybe it was an earthquake. Maybe it was a volcanic eruption. Now, of those three hypotheses, which one is best? Well, according to Lyell's principle, the best explanation is the one that invokes a cause which we know from our experience can produce the effect we're trying to explain. So with that criterion in mind, the best explanation is clearly the volcano, because we've seen volcanoes produce white powdery stuff, and floods and earthquakes don't do that. And so that was the mode of reasoning that I learned about from studying Lyell and Darwin. And then one day I realized that applied to the question of the origin of information. And that was the key question that was fascinating me. Because I asked myself this question: what is the cause now in operation for the production of digital code? What is the cause that we know of that generates code or information? And soon after that, I came across something else I was reading. It was a little book that was an application of information theory or information science to the analysis of DNA. And it was written by a pioneer in this field named Henry Quasler. And he made an offhand comment, as it happens, on page 16. I remember vividly the day I came across it. And Quasler said that the creation of new information is habitually associated with conscious activity.
Key phrase.
Key phrase, because for Lyell, it's our uniform and repeated experience that teaches about cause and effect. Mm-hmm. So if something is habitually associated with conscious activity, if information is habitually associated with conscious activity, it really fulfills Lyell's dictum that what we're looking at is a cause now in operation, a cause that routinely produces the effect in question.
In fact, that's the only place we've ever seen it come
Well,
from.
that was the next thing. As I began to think this through, I thought, whenever we see information and we trace it back to its source, whether we're talking about a computer program or a hieroglyphic inscription or a paragraph in a book or information embedded in a radio signal, whenever we see information, we trace it back to its ultimate source, to its ultimate cause, we always come to a mind, not a material process, so to conscious and rational activity, just as Quasler said. So I realized that his little statement here was absolutely right, and it actually formed the basis of a scientific case for intelligent design using the method of Darwin and Lyell. And so in my first book, Signature in the Cell, I developed an argument for intelligent design, self-consciously using the method of Darwin and Lyell to show that the origin of the information you needed to build the first living cell was best explained by an intelligent agent, by the activity of a designing intelligence in the history of life. But I think the same reasoning can be applied to explain this mysterious event we call the Cambrian explosion, because as we've been saying, the Cambrian explosion is not just an explosion of new biological form and structure, it's an explosion of information, both genetic information and then this higher level information that we've been calling epigenetic information. In both cases, we know of only one kind of cause that produces information, and that causes intelligence. And insofar as the Cambrian explosion is an information explosion, I think we have another clear example of an event in the history of life that is providing decisive evidence for intelligent design.
Yeah, I loved the first illustration that you had, I think, in that book, where you had a picture of a heart that was drawn on the sand next to the ocean. And it said, "I love you, Joanne." And if you came up and you're walking down the beach, you wouldn't assume that that heart and that message in there was the product of water just rolling up against the sand, okay? We immediately see a message, we got information, and we say, someone, intelligence, created that message.
Well, we use this kind of reasoning all the time, and it's precisely because of what we know about the cause and effect structure of the world. Bill Gates has said that DNA is like a software program, but much more complex than any we've ever created. Well, we know from experience that it takes a programmer to make a software program. And we know that generally, that whenever we see information, especially when we find it in a digital form, there has always been a mind at work in producing that information. So when we find information at the foundation of life, when we see evidence of big infusions of information in the history of life, I argue that we're seeing decisive evidence of the activity of a designing intelligence, because we know of no other cause in the whole range of our experience that is sufficient to produce information in that kind of a form. And we have, in fact, as we've been talking in previous programs, we also have very strong reasons now for doubting that the Darwinian mechanism can build both the genetic and the epigenetic information necessary to build the complex animal forms that arise in the Cambrian. So we have both a negative case against Darwinism and other similar materialistic evolutionary theories. And in my book, I show that it's not just the neo-Darwinian evolutionary model that fails to account for the origin of information, but the other newer models that have been proposed as well. And on the other side, we have positive evidence, positive experience of a cause and effect pattern in the world where we see that it is a mind or an intelligent agent which invariably is responsible for the generation of information. So I think there's a very powerful case to be made for intelligent design, and the ironic thing, the somewhat mischievous thing, if you will, is that in formulating this case, we've made the case for intelligent design using the same method of the same standard method of historical scientific reasoning that Charles Darwin himself pioneered in The Origin of Species.
Yeah, you made an important statement in your book, and I want to put this up on the screen for folks. You say that just because the Cambrian explosion may exhibit some features for which intelligent design is a known cause, does not mean that intelligent design was necessarily the actual cause or the best explanation of those features. You go on to say, only if the Cambrian event and animals exhibit features for which intelligent design is the only known cause, may a historical scientist make a decisive inference to a past intelligent cause. I think that's a very important statement. Explain what you're saying here.
Right. Well, this is a point of logic. And when you're trying to do what scientists call retrodict, infer from clues in the present back to causes in the past, you can make a decisive inference when there's only one known cause for the effect in question. If there are many different types of causes which will all produce the same effect, then when you see the effect, it could be any one of those causes that was responsible. But in the case, for the historical scientists, the best case is when you have an effect for which there's only one cause, because then the effect will decisively indicate the cause because there's no other possibility. And what I argue in Darwin's Doubt and also in Signature in the Cell is there is only one known cause for the origin of functional information, information that performs a communication function, and that cause is intelligence. And yet that's precisely the effect that we see in life, that we have to explain in life. It's the origin of the information in the digital code in DNA, and then we've also been talking about this higher level information processing system and the epigenetic information that's involved in building animal body plans.
All right. I think we're going to show them another clip here that's going to explain not only what you're saying, but give them a little bit more evidence about why intelligent design is the best explanation to fit the evidence. Folks, I want you to watch.
As I've reflected on this over the years, I've realized that the same reasoning that applies to the origin of biological information also applies to the origin of the other key features of the Cambrian explosion.
The sudden top-down appearance of the phyla during the Cambrian explosion defies the simple to complex pattern of development that Darwin predicted.
In Darwin's picture, you'd have little differences accumulating to big differences. The top-down picture turns that on its head.
You find first you get new phyla, and then you have some variations on those themes over time. But the new form, the big differences, appear right from the beginning in the fossil record. If you consider the possibility of design, you realize that that pattern makes perfect sense, because we see in our own history of technology this same pattern of top-down appearance in new forms.
You're always working from your high-level objective to your details in order to accomplish the high-level objective.
Only intelligence can visualize a complex endpoint and bring together everything that's needed to actualize that endpoint.
The body designs evident in the Cambrian animals have continued to appear in different species throughout the history of life. Yet though these species share common body plans, they are not connected by a continuous line of intermediate material forms.
The continuity that explains that consistency of form through time is the continuity of an idea. So when we see in the fossil record the same basic idea popping up over and over again, that suggests that a mind has played a role in the origin of that form, of that body plan organization.
Design systems also display another distinctive feature. They are comprised of a network of complex and precisely organized component parts.
You could speak of these in terms of a nested hierarchy. You have the very high level parameters that specify the whole project goal. Below that you have layers and layers of more detailed parameters that are needed in order to complete the whole project.
As an example, resistors, capacitors, and transistors are each made from specifically arranged materials. These components are then assembled to form integrated circuits. Circuits are arranged to build computers that are then arranged into networks of computers.
So at each level there is a specificity of arrangement that's provided in turn by the intelligent designer, the engineer, that keeps the whole system working. Well, what you have in biology is something very similar.
In living systems, genes code for proteins, which are organized to form distinctive cell types, which are arranged to form tissues and organs, which are assembled into body plans, including the plans that arose during the Cambrian explosion.
We know of only one cause in the entire universe that can produce that kind of hierarchical arrangement of form and structure and information, and that cause is intelligence. This is the kind of thing that minds do, but natural undirected processes don't.
Evolution works very slowly, as Darwin saw it, with lots of failed experiments along the way. And one would expect that over millions of years, as sediment is being deposited, that you would capture some of those experiments, some of those linking groups leading to the trilobites that he knew all about.
So the absence of those forms is profoundly mysterious. But from the standpoint of intelligent design, it's not mysterious at all, because we know that intelligent agents can bring things into existence that didn't exist before because they had an idea. They had a blueprint in their minds that they realized in their creative activity. There's no need to tinker through millions of years of evolutionary history if you can actualize a plan at a discrete moment in time. And that's exactly what appears to have occurred in the Cambrian explosion.
In brief, Stephen, I think you've made a really impressive case. What do you think are the implications of this case?
Well, I think one of the first things that is striking to a lot of people when they hear this is how different intelligent design is from the way they've had it presented in the media. And we've been told over and over again that Darwinian evolution answers all the key questions, and yet we're finding in the top-level evolutionary biology journals that many leading scientists are acknowledging real problems.
So why do you think all this is so important?
Well, I think it's important first scientifically because we've heard, for 150 years that there's no evidence of design in nature, only the appearance of design. Richard Dawkins has said it well, biology is the study of complicated things that give the appearance of having been designed for a purpose. But according to the Darwinist and the modern neo-Darwinist, that appearance is an illusion. And I think what we're actually seeing is compelling evidence for real design, for the activity of an actual designing intelligence in the history of life. And that's a major reversal in 150 years of scientific thinking. But I think this discovery also has larger worldview implications, because every worldview has to answer the question, what is the thing or the entity or the process from which everything else came? And Darwinism has played an indispensable role in providing support for what's known as a naturalistic or materialistic worldview that says that everything came about as a result of undirected, unguided material processes. And if the theory of intelligent design is correct, and I think there's strong evidence for it, then I think the materialistic worldview, then I think that this evidence is posing a major challenge to the materialistic way of thinking, and that we may have scientific discoveries that have larger theistic implications that are pointing to the need for a designing intelligence that indeed has the attributes that Jews and Christians have long explained the origin of information. For example, there's a wonderful new evolutionary theory known as natural genetic engineering, and it's been put forward by a really top cell biologist at the University of Chicago named James Shapiro, someone for whom I have great respect. And Shapiro has shown that the mutations that Darwinists rely on to account for biological change are not actually random at all, but they instead seem to be under what he calls algorithmic control. There's a pre-programmed adaptive capacity in organisms that allows them to respond to various kinds of environmental stressors. And I think that's fascinating, and I think he's made a very good case that these types of directed mutations are occurring in response to various types of environmental challenges. But the question that Shapiro doesn't attempt to answer is the question of the origin of that pre-programming.
Where did it come from?
Where did that come from? And that, again, is an information question. And so I show in these later chapters in the book that, yes, there are new models of evolutionary theory, and that in part documents my claim that neo-Darwinism is dead. The standard textbook theory is no longer accepted by leading evolutionary biologists themselves. They're moving on to try to develop new mechanisms to explain the origin of form and information. But I also show that these new theories invariably either don't address the key question of the origin of biological information, or if they do, they simply presuppose prior unexplained sources of information and therefore leave the question unanswered as well.
Yeah, it seems like they've got a brick wall at the beginning and at the end. They don't know how to get the information, and once you have the information, you can't mess with it very much.
Well, we found that was true as well. In an earlier segment, we talked about the problem of these developmental gene regulatory networks, how these networks of genes full of information are critical to building animals and animal body plans in the right way, and yet you can't perturb these networks without destroying the process of development. So,
yeah, we've been talking about some of the critics here of your book, but there was one paleontologist that actually offered a suggestion or solution, a theory.
The key argument of the book concerns the origin of the information needed to build the animals. And finally, in September of 2013, the year the book was released, a review came out with a reviewer who attempted to address that key issue. And this was a review that was published in Science. It was written by, Science is the largest science journal in America, and it was written by Charles Marshall, a leading evolutionary biologist and paleontologist at the University of California, Berkeley. And Marshall's review was critical, and yet also, I thought, respectful. And it was respectful in an important sense that I found a relief. He attempted to address the main argument of the book. And he said, look, Meyer's case depends upon the claim that the origin of new animal body plans requires vast amounts of novel genetic information. In fact, this isn't our present understanding. Our present understanding of morphogenesis, which just means body plan building, indicates that the new animal phyla were not made by new genes, but largely emerged through the rewiring of gene regulatory networks of already existing genes.
Yep.
Now that's a really interesting passage, because he is attempting to refute the argument I'm making about the need for intelligent design to explain the origin of information. But he doesn't really explain the origin of information. He presupposes pre-existing sources of information. Number one, he presupposes the existence of gene regulatory networks, which we saw in a previous episode, are networks of interacting genes and their protein products. Well, all those genes contain genetic information. Plus, we know these gene regulatory networks act on other pre-existing genes to turn some on and turn others off at different times. And so that's another source of pre-existing genetic information. And thirdly, rewiring the genetic regulatory networks requires requires changes of code, more input of information, and in all three cases, Marshall doesn't explain where this information comes from, he just presupposes it. So I don't think you need a PhD in biology to see that he hasn't refuted the argument I made. Instead, he really begs the question. He just puts the problem of the origin of information back a little bit and says, well, there's all this pre-existing information that explains how the animals develop. In addition to all that, he suggests that the evolutionary process could rewire and alter these gene regulatory networks. And as we discussed in a previous episode, one of the things we know experimentally is that you can't perturb these networks of genes and expect animals to develop properly. Instead, if you alter these networks of genes, they're going to die. And so that's actually not even a plausible suggestion based on what we know experimentally.
Yeah, I also have to say that there are a lot of scientists that made some positive statements about the book. I mean, I read the one professor from the Max Planck University or Institute in Germany who said that it's the most accurate, comprehensive review of the scientific literature that he's seen in 40 years. I mean, what did that do for you?
Well, we were really pleased with some of the endorsements for the book that came in from, there was a Harvard professor of genetics, a leading Cambrian paleontologist who's published a book on the Cambrian explosion with Columbia University Press, this Professor Lunig from the Max Planck Institute in Germany. So yeah, we've had supporters as well as critics.
Yeah, let's cruise on with some of the other objections that we hear a lot of times in the popular press, and they'll say that intelligent design is not science.
This is a very interesting objection, because what you want to know about an idea is whether or not it's true or false. You don't want to know how to classify it. Okay, so if we say that there's strong evidence that a designing intelligence acted in the history of life, and someone comes back and says it's not science, that's kind of an odd response. What they ought to say is, no, that's not true. The evidence points in the other direction. And rather than disputing the factual claim as being untrue, people have said, well, no, it's not science. It's religion, or it's philosophy, or it's pseudoscience, or it's something else. And so the first thing I'd say about that objection is it's really not on point. What we want to know is whether the theory is true or not. Now, I would also say there's every good reason to regard the theory of intelligent design as a scientific theory. As we discussed in a previous episode, in formulating the case for intelligent design, I self-consciously used the same method of scientific reasoning that Charles Darwin used in The Origin of Species. And so if you want to say that the theory of intelligent design is unscientific, you would have to say that Darwin's theory of evolution by natural selection, which he justified using an historical scientific method, is also unscientific. So I don't think actually the attempt to classify it as unscientific actually works, but it is kind of an odd claim. The issue is whether intelligent design is true or not, and I think we make a good case that there's good reason to believe that it is providing us the best explanation and most likely to be true.
Yeah, tell the folks about what we said last week, for those that missed it, about looking at Darwin's mentor, Charles Lyell, and then why is this a historical scientific theory?
Right, well, the theory of intelligent design and Darwinian theory, or various materialistic evolutionary theories, are both trying to answer the same question. How do these complex forms of animal life arise in the history of life? What caused them to come into existence? So they're both
in the past.
They're in the past, in the remote past. So they're both causal theories about events that happened in the remote past. And intelligent design is, the theory of intelligent design is affirming or inferring that there was an intelligent cause that played a role in the origin of the information necessary to build these forms of animal life. Neo-Darwinism and other materialistic theories of evolution say the opposite. Those theories are saying, well, an undirected material process produced the form and information that we see arising in the history of the past. These are two competing explanations of the same phenomena. One isn't scientific and the other religious, or one philosophical and the other pseudoscience, or something like that. They're not two different types of things like that. They're not two different types of things. They're two different answers to the same question. So if one is scientific, the other is also scientific, but providing a competing scientific explanation.
Yeah, I think that's something that's very important for people to understand. Let's go back to the point that intelligent design is not a testable theory. I'm thinking more and more that that's not true, because you're testing it all kinds of ways.
Well, that's right. One of the reasons that people say intelligent design isn't science is they say it's not testable. But they have a very narrow and inappropriate notion of testability, and that is that you must test things in a laboratory under controlled laboratory conditions. But that's not how you test historical scientific theories. Historical scientific theories use a method of reasoning known as the method of multiple competing hypotheses, where scientists propose multiple possible explanations for an event in the remote past, and then they test the credibility of those hypotheses against our knowledge of cause and effect in the present. And that's exactly what intelligent design as a theory does. It proposes an intelligent agent acting to generate information as the explanation for the origin of information that arises in the remote past. It then tests that idea against our knowledge of cause and effect and shows that there's only one known cause of the origin of information in our present experience, and that cause is intelligence. So intelligent design meets the key test of what's called causal adequacy. It proposes a cause which is known to produce the effect in question. And there are other ways that you can test historical scientific theories as well, and I show in my book that there are actually 3 separate types of tests, and the intelligent design is testable in each of those relevant ways. And so it meets the test of, or it meets the criterion of testability, and there's therefore every good reason to regard it as a scientific theory.
Okay, and then the big one is that people say, hey, intelligent design is religion masquerading as science.
Right, and I think the idea that intelligent design is religion, not science, confuses the basis of the theory of intelligent design with the possible implications of the theory. And the basis of the theory of intelligent design is the scientific evidence that we've been looking at. The sudden appearance of the major groups of animals in the fossil record, the evidence of digital information stored in the DNA molecule, and many other classes of evidence that I talk about in my books. So the basis of the theory is evidential. It's the evidence of the natural world, and also the use of a standard method of scientific reasoning, the very method that Darwin used. So the theory is based on a standard method of scientific reasoning and based on empirical evidence from the natural world. Therefore, it has a scientific basis. It may have larger worldview or philosophical or religious implications, but then again, so does Darwinism. Darwinism has larger implications that support a materialistic worldview, as many supporters of Darwinian theory have noted. And the theory of intelligent design may also have larger worldview or philosophical or even religious implications. If we see evidence of a designing intelligence in the history of life, that's consistent with what the theistic religions have said. They have affirmed that there is a designing agent that's responsible for creating the life forms we see around us, and they affirm that that agent is God. And so while the theory of intelligent design does not prove the existence of God, it affirms a tenet of the theistic religions, which say, yes, there was a designing mind, that mind is God, and so you can say that the theory of intelligent design has implications that are theistic friendly, that affirm a key idea that is affirmed by Jews and Christians and other people who believe in God. So that doesn't mean the theory is based on religion, but rather it has implications that are friendly to religious belief.
Yeah, talk about the verse that's over the door at Cambridge University, Psalm 111.
Well, I've made this clear in a lot of my public discussions of intelligent design. Intelligent design, again, is based on scientific evidence and a scientific method of reasoning, but it has larger implications that are friendly to faith. I happen to also be a person of faith. I'm a Christian, and one of the things I found inspirational when I was doing my PhD work in Cambridge was an inscription that had been placed on the door of the Cavendish Laboratory by the famed 19th century physicist James Clerk Maxwell. Is one of the, I think, arguably one of the three great physicists in the history of science. And Maxwell was a devout Christian who wanted the inspiration for science to be publicly proclaimed. And the verse that he had inscribed on the door of the Cavendish Laboratory captured that in his view, and it was from Psalm 111. It said, "Great are the works of the Lord, sought out or studied by all who take pleasure therein." And for him, this was the inspiration for doing science, is that he believed the natural world had been created by God, and it had been made intelligible to the human mind because God was a rational creator, and he made our minds in a rational way so that we could understand the logic and design that he had built into the creation.
Yeah, I like the next part down there. He has caused his wonders to be remembered. What does that mean?
Well, when I sign my book to people who ask for a signature, and I often put that reference to the Psalm 111:4. It says, "He has caused his wonders to be remembered." And I, again, as a person of faith, find this kind of inspirational, because I think what we're looking at in the preservation of fossils, in our discovery of the DNA molecule, in the cosmic background radiation coming from the early echo of the beginning of the universe, I think we're seeing indications of the activity of the designer who made the world around us, and that we can discover these indications long after the fact of the events that we're studying, I think is pretty exciting. And it's kind of interesting that the psalmist indicated that such a thing might in fact be possible, that God has caused his wonders to be remembered.
Yeah, I love it. I want you to say an encouraging word to students who are going through school, grad school, PhD work, and they're in turmoil because of what they're being told on one side about how evolution impacts their faith, and now they're hearing this information. You can remember struggling with these issues. What would you say to encourage them in terms of following the evidence wherever it's going to lead them?
Well, I do have a lot of conversations with young scientists, grad students, students in their undergraduate years, and if they're skeptical of Darwinism, or if they have a religious faith, if they are theists of some kind, Jews, Christians, other, they will often feel that that they're the odd man out in their science classes. Many professors hold a naturalistic or materialistic worldview. The professors often think and will say openly that science properly understood supports such a worldview, or many of them are openly atheistic. We'll say, you know, like Richard Dawkins believes that the science properly understood supports an atheistic perspective. And so students in classes with professors who are overtly opposed to any kind of theistic belief will often feel kind of intimidated and feel like maybe they're the person that didn't get the memo. And I think actually that this is a tremendous time to be going into science if you're interested in the big questions, and in particular if you have a theistic perspective, because I think we're seeing evidence of design not only in biology but also in physics and in cosmology. And I think that we often get the opposite perspective. You know, you have the science popularizers are going to say, you know, science supports the new atheism, or science makes belief in God untenable, or things like that. But when you dig into the evidence, I think there's a powerful case for intelligent design, and I personally think that the evidence doesn't just point to a designer generically, but instead there's a powerful case to be made from the evidence in biology, but also in physics and cosmology, for a designer that has the same attributes, the very attributes that Jews and Christians have long ascribed to God. From the biology, I think we see the evidence of a designing intelligence who's acted in the history of life. From the physics, I think we see in what was called the fine-tuning evidence of design from the very beginning of the universe. And in cosmology, I see evidence of a definite beginning to the universe that seems to point to the need for a transcendent form of causation, a cause beyond matter, space, time, and energy. And so I think when you add all that up, I think there's a very strong case for theism to be made. And you and I have talked about that in a previous series of interviews where I make that case in more detail. So right. I think rather than science properly understood undermining faith, I think it actually provides evidence for faith, maybe not a proof in the logically certain sense, but I think strong evidence that is very friendly to theistic belief. So I think it's a wonderful time to be a person of faith and a person who's interested in the natural world. And so I would really encourage students to get into science and also find out the other side of the argument. You know, the universities are still dominated by a class of professors who are typically going to be more of a naturalistic or a materialistic mindset. But there are a lot of really good scientists and philosophers of science who are looking at the questions in a different way and who are making a case for a more theistic-friendly understanding of science. And so dig in. It's an exciting time to be interested in these questions.